Hello everyone, thanks for coming to my talk. I'll warn you that this is my first public talk that I've given, so I might have a tendency of speeding up, and at some point I might have to go, right, I've gone too fast, and stop for a bit. But feel free to stand up, wave your hands around if I'm going a million miles an hour, and basically I hope that you can get more out of this rather than going, he just kept going and it was really, really fast. So I might as well start. As my talk suggests, marine life of the Northwest. I won't be talking, explaining too much on the breakdown of animal groups or anything like that. It's more talking about the Woodside partnership with the museum, where Woodside has been kind enough to sponsor us in biodiversity research. They've been a principal sponsor for the last 10 years for our field trips. And I'll be just talking about the results from these expeditions, a bit about why they're important. But firstly, I'll end up talking about the museum collections. The first question I'll get if I don't go through this is why bother having the collections. So I'll be talking about what we've got in the collection and why we have it, how's it used, what's the point of keeping all these specimens in jars, does anyone else see them, or are they all just locked up in a cupboard? And then I'll get into the Woodside grants, talking about just a brief talk about how we go about actually collecting the specimens, what we do with the specimens in the field, and then I'll be giving some results about the completed surveys, and then giving preliminary results for the current project that we've got going with Woodside. So the collections. Now, our most famous specimen will be the 23-meter blue whale skeleton, which washed up in the 1890s in Bustleton. The Bustleton Township charged the museum £25 for the right to take the skeleton from the beach. And then, excuse me, they then carted it up by horse and cart all the way up to Perth, where it was put together. Oh, I went too fast. What do I do? All right. Anyway, that's how the skeleton would have looked, should look, and that's how it was for many years on the 5th floor of our Francis Street building in Perth. Unfortunately, since that building's been condemned, we've had to take it down, and it now looks a bit like this in our collection centre. We hope and pray one day that we'll get a building that's big enough to hold a 23-meter skeleton again, and so that way we can put it back together and restore it to the way it should be for the public. As you can see, under wraps, it just looks a bit sad. Fortunately, in my section, the rest of of aquatic zoology, the rest of the specimens are much, much smaller and fit handily into jars and fit on these shelves. For the aquatic zoology side, we have 640,000 specimen lots. Now this is effectively just the number of jars. These could be wet or dry. For the crustaceans, we tend to keep things wet. If they dry out, they tend to get a bit brittle and fall apart. Now, the major major violator in terms of raping and pillaging and keeping things in our collection are the mollusks. The joke is that they keep everything, from the tiniest shell fragment to full giant clams. So then there's the fish collection. Once again, they've got a long history with of having their own section within the museum. Ray George, who's here tonight, is actually the first curator of Crustacea. I think he was actually the curator of all marine invertebrates for a long time as well. A large portion of the crustacean collection, of course, was collected by Ray from when he started. And basically, to give an idea of what sort of scale that we've got, other than just sheer numbers of jars, our scope is to document the Western Australian fauna. We do have species from outside Australia, New Zealand, Madagascar, Europe, and that's basically the way we break it down. These numbers, quite hard to get any sort of scope of what those numbers mean, but the 2,700 species that we've got of crustaceans in the collection, it represents a bit over a third of the total number of species of crustacean species in Australian waters, which is good, seeing as Western Australia takes up about a third of Australia. So we've got a fairly good coverage. There are gaps, and we're always after more specimens. So why do we actually keep these? Of course, they have great scientific use. First and foremost is the taxonomic use. This is the description of species, the classification into the Linnaean system of nature. So we collect our specimens, we compare them we hopefully find new ones, we describe them, and among these, the most important specimens in our collections are the type specimens. Now these are the original specimens used to define the species concept to which the genus and species, the scientific name, is attached. So whenever someone says that they caught a blue manna crab from out of the estuary, they're effectively comparing it back to the original species description by, I think it must have been Linnaeus actually that described that one, back in 1767. That's the idea. The species concept isn't set in stone. People have different opinions about what constitutes a species. It's a bit of a minefield, and you always hear, well, the hairy marron is a good example of where people have mismatched the ideas are different. I don't know how close to that is to being properly resolved, but the hairy marron versus smooth marron in the Margaret River, very contentious issue. And I have it on good authority that Portunus pelagicus, which is the common blue manna crab, might be getting a name change soon too, which is going to upset fisheries quite a bit, because one thing that people don't really realize is that the taxonomic aspects of things are actually written into legislation for commercial species. Like wherever you've got something under quota management, they're referring to a species or a group of species to which they're saying, right, that's what you're catching. If it fits within these boundaries, that's the species you're catching. Therefore, you can't catch more than so many or that size or whatever. So when something changes in the taxonomy that affects something in the legislation, things get awkward. But also, outside of that, we also gain valuable biological knowledge about individual specimens as well as the species, reproductive habits, ages, sizes. Fish collections are becoming increasingly sought after to gather information about ages of fishes at maturity. And basically one of the ways they do this is actually to take out the air bone of the fish called the otolith, and basically that, much like a tree trunk, has growth rings. You can count the growth rings, get the age of the fish, check the reproductive status, whether it's mature, female, carrying eggs, things like that, engage the age at maturity, and then you can start talking about, right, how much bigger do we need to get them before, so that they've got at least some time to spawn before we can catch them for fisheries purposes. One story that I got told back in New Zealand is the orange ruffy species of fish, very old, slow growing, deep sea species. You can actually date, basically one of the ways they calibrate the dates of the otoliths is that they can detect when the Hiroshima bombs were dropped in the otoliths. That's how they know how old. Basically there's a point in any calcium deposits built over the last 60 plus years when Hiroshima dropped. So very valuable. And of course this applies to paleo as well, where you start looking at paleoclimate, the prehistoric time, things like that. The distributions, very important also for fisheries management. You need to know where your species are if you're going to try and catch them, and also know that if you're fishing heavily in, say, Cockburn Sound, that there's going to be more outside of Cockburn Sound. So the distribution's geographic as well as bathymetric, so the depth, and also native and introduced species. I've noticed you don't have sparrows here, but you've got them on the east coast. No koalas, which annoyed me. I thought they were everywhere in Australia. Same with wombats. But kookaburras, as much as Western Australians don't seem to like them so much, I'm quite happy to hear them. It's an iconic Australiana for me. Expect them to be everywhere, but no, they were actually introduced. A lot of people, well, why would you suspect it to be introduced? Same goes for the marine stuff. Everybody thinks that the water goes all the way around Australia, except for when it gets too hot for a species. Surely the species are found all the way around. But no, you find that Australian bite, you'll get species on either side that are different, very similar species, but different on either side. Same with across the top as well. Ecological, now this is more talking about the individual species ecology as well, but also more of the ecosystem itself. So the community structure, when we go out on our surveys, we collect a lot of specimens from the same spot, from a broad range of taxa. We can build up a picture about what the environment looks like from a biological point of view. But you can also get things like the diet from the gut contents of the collected species. Now the molecular side of things, this is more of a quite modern approach for collections, or application for collections, sorry. Through preserving the DNA using ethanol, we can do genetic tests to determine species. There's a lot of, as I said, there was a bit of dispute about what constitutes a species. DNA is now moved in as being another tool in defining where species boundaries lie, and also bioprospecting. Now, the term bioprospecting is reasonably new, but it's something that humans have been doing since we realized that herbs have certain medicinal purposes. Now, today, though, it's more anti-cancer agents, and to some degree, unfortunately, cosmetics as well. Whales, sharks, all were heavily hunted for various cosmetic agents, as well as oils and bits and pieces like that. Today, focus has shifted from the large animals into the smaller animals, but still, now the collections are being actually looked at as a way of first testing out before we actually go out into the environment and actually effectively go hunting again. Educational is the next step. Of course, the museum is an educational institution as well as research. We use our specimens for exhibitions. We try to cycle these through so that as they get damaged or if they get old, we can replace them with fresher ones, but sometimes doesn't always happen when we've got particularly rare things on display. We also produce guides, either through these one-page pamphlets or through various books. We've got a reasonably large publications team that we've got our own records journal, but we also produce guidebooks, and the latest one is the Sea Stingers should be hitting the shelves soon, hopefully, a revised version of the Sea Stingers book. But outside of this, the scientific use all can sound a bit esoteric, hard to understand how this affects anyone else. But this filters through, as I said, with the taxonomy, it gets put into legislation, making things very, very, very official for us. And then, of course, fisheries management is important for them. They need to know what bycatch is coming up with prawn trawls. They also need to know what sort of damage they might be causing to habitats. Environment, and that's a subset of environmental management, I guess, as well as the next one with risk assessment. We're getting increasing amounts of requests for identification of specimens collected from in and around mine sites. I prefer to stick with the marine things, but I'm starting to get a lot of slaters come in. Basically, the idea with the mine sites is like target groups tend to be, some of the trapdoor spiders tend to be quite large, quite high on the consultancy's list of animals that they need to get specific identifications. Basically, seeing as a trapdoor spider spends all its time in its burrow, the eggs hatch into juvenile trapdoors. They disperse a little bit, build their own burrow, build the trapdoor, stay there for the rest of their lives. They're considered, in technical terms, as an SRE, which is a short-range endemic, meaning that a particular species might only be restricted to a certain hill or a certain valley, and not particularly well dispersed. So instead of ranging all across the Great Sandy Desert, it might only be found in the southern part, the central part, eastern part, western part, depending on how it goes about its business. And unfortunately for me, slaters are also considered these short-range endemics, and I'm under increasing pressure to learn what they are. Also conservation, which basically should be sitting at the top of the rest of these ones in terms of priorities. And tourism. The reason why people go to the Great Barrier Reef is because people realize there's a lot of things to see. And who told them there were things to see? We did. Well, maybe not Western Australia Museum, but you get the idea. Same with Ningaloo. We are pointing out areas of high diversity. The Stirling Ranges National Park? That wouldn't be a national park if it wasn't for its great diversity of life. The stat that I hear is that it's got more flowering plants than Europe, or at least Northern Europe. Even though it's half the size, which is staggering. But yeah, so that's why we have the collections. In terms of why do we keep them for so long, instead of just publishing our results and saying, well, we saw them there from these places. Basically, as I said, the species concept is fluid, reasonably fluid. So basically, having the specimen is the final say. It's like, right, that's what we call this. We've got it here. That proves that what that was there, assuming the associated data is correct, of course. But we like to think it's always 100% correct. So after talking about the environment, it shouldn't be anybody surprise to anyone that there is an increasing demand for Western Australian resources. Quite clearly, this is something you all know. And the recreational and tourist industry, which is something that people don't really quite understand as having an impact. Obviously, mineral wealth, you dug a big hole in the ground. Clearly, that's going to have an effect. And with some of the chemical processes for mining, such as the use of cyanide to extract gold, that's going to have an impact. Large fisheries, also. Clearly, we need to start thinking about what's actually happening when we're carrying out our various activities. So as a result of the increasing demand, you've clearly got an increase in the pressure on the system. I don't need to preach that pollution, habitat destruction, and overfishing are going to damage what we've got in the environment. We need to make sure that what we do, the environment can actually sustain the activities that we want to carry out. Recently, the Marine Stewardship Council, basically the Western rock lobster, was the first green-listed fisheries in the world by the Marine Stewardship Council, which basically assesses the fisheries methods used, population stocks, things like that, to gauge whether or not something is actually an ethical fishery. Western rock lobster has subsequently gone under a little bit, much to fisheries' disgrace. But yeah, even what we think is quite good can all of a sudden have a dramatic change. Basically, there's so many things we don't know. We got no real way of predicting things. There's been a few theories about what happened to the rock lobster. One that I quite am leaning to is that with the Perth Canyon, there's an eddy or vortex going around. Oh, sorry, off the coast of Perth, just off the back of the west coast of Rottnest. Basically, with the Swan River, over prehistoric times, has dug a channel down through the continental shelf just off the coast. And basically, the Perth Canyon, I forget the figures on how on the depth or breadth of it. Yes, it's dark and it's cold. Effectively, in 2006, some oceanographic monitoring showed that there was a vortex, basically a big whirlpool caused by the Lewin current and some other factors that I don't really understand myself. And basically, that was labelled as a death trap for any fish larvae. Basically, they would get sucked down, and if the fish larvae aren't strong enough swimmers, they couldn't stay at the surface where they needed to stay to feed on plankton. And I wouldn't be surprised if all the lovely little delicate phylosoma larvae of the crayfish, I don't know if you've ever seen pictures of them, but they look like aliens. They don't look like crayfish at all. They've got a big round head and big feathery legs, and they just would get sucked down. And if they get too deep, gets too cold, they might not be able to make it back. And you wouldn't notice that for years. And so 4 years later, I think the coincidence is a little strong. But anyway, back onto that. That was a small diversion. Need for basic environmental data is quite clear. Not just biological, but you could go through the entire list of the the physical sciences, and you can say, we don't know enough. And to be honest, I don't think we'll ever know enough. We'll always find some way of going, oh yeah, maybe we need to know about that as well. But Woodside seem to have come to the conclusion that they need to take a proactive role in environmental research. And so they've been kind enough to sponsor us on some biodiversity trips of our own. First of which was received the most funding and is by far the largest trip that we've done in recent years, all before my time, unfortunately, to the Dampier Archipelago. Obvious site of industrial activity. You've got a lot of mineral iron ore shipped out of the ports nearby. You've got Barrow Island just to the south, which is another big iron ore facility. You've got huge gas deposits up around here that are about to effectively send us into the next boom. Sorry, I'll get rid of that. And then after that, once we'd finished that off, we went to the Northwest Atolls. Now this covers some very sparse, a very broad area of the Northwest Atolls. We went sampled Mermaid Reef, which is the southern, well, the middle dot now, which is part of the Rolly Shoals shelf reefs. Now, these are offshore atolls. And then, I've clicked too far, but the current trip that we went on last year in October, southern Kimberleys, and now we're going on to the northern Kimberleys, hopefully this October, hopefully. Now, our aims for the museum is, well, basically it's in our charter, I think, to actually in the Museum Act and the legislation to document Western Australia's fauna, and that's what we try and, that's our aim whenever we go out. We've always had staff with expertise in crustaceans, well, maybe not always, but traditionally that's our strengths, is crustaceans, the echinoderms, mollusks, fish, and more recently sponges. Our head of department is an international sponge expert, and we'll be giving a bit of a talk about what sponges actually are a little bit into this. But when we can, seeing as a lot of these areas are coral reef zones, we always take along someone that knows something about corals, or else we're just looking at, well, to us it's just living rock. And also we try and make sure we've got someone that can look at the flora as well. We don't want to be labelled as being one-sided in what we're what we're trying to achieve. And when space or money is available, we'll take on other groups, such as the marine worms, yeah, and a few other bits and pieces where we can. I'd like someday for us to be doing some of the planktonic work as well. Diving, I'm onto getting into the methods side of things. Basically, diving is one of our mainstays of collecting these days. And for us, well, for me, I follow a transect line, which basically is a tape measure rolled out 25 meters or 50 meters, depending on what we're on. And I basically surveying a meter-wide strip next to it. That's a gauge on how far I've gone. And I do a meter-wide strip, and I'm turning over rocks as I go, picking up and bagging bits of seaweed, dead coral, and whatever else I come across. I'll explain why I pick up bits of coral and other bits and pieces as I go along. There's usually quite a lot of us following this transect line at one time, so we try and stagger ourselves. But once we've done our transect, each group has their own little methodologies and quirks. The fish in particular tend to get down on the buoy line where we've set our station and go, right, well, we're going to take a random bearing and get away from everybody else, because we do tend to stir up the water and scare off fish as we go through everything else. Diving is not always straightforward. It's not just following a line coming up to the surface at the other end. Seeing as we are stirring up the bottom, a lot of food for the fish gets flicked into the water column. They know we're picking up bits of food that they would like to have. And this particular potato cod actually quite liked going after a Mel's catch bag, which was full of bits of crabs and shrimp. And apparently it took after 5 minutes of trying to fend this thing off, and it just kept coming back and just biting on the bag, pulling on it. One of the other divers apparently came up with the shark shield and gave it a bit of a zap, and it took off pretty quickly after that. We also have dangerous animals. Sea snakes aren't to be sniffed at. I know a lot of people say that the mouths are too small, they're not going to bite except for anywhere between the webbing and webbing of your fingers and things like that. If you've ever seen a sea snake eating a fish on the surface, their mouths get big. Snakes are known for dislocating their jaw so they can bite things. So they're usually quite curious and will see what you're doing, but I'm sure if you poked it too often, it might swing around and get you. Now, when the tide's out, we also like to get to the reef. For me, this is quite a lot simpler exercise in collecting. I can pick up a rock out of the water, and when it comes to things like the swimming crustaceans like shrimp, it's a bit harder for them to get away from me. And also underwater, a lot of shrimp are clear. I can't see them anyway. And things are easier to chase outside of the water. If they are getting away from me, I can move a little bit faster and a bit more agile on the reef than I can underwater. And crustaceans are notorious for being just a little bit quick and darting all over the show. Mangroves, they're a scary piece of habitat to actually work in. If you get it particularly dense, it's quite dark, it's very still, humid, and there's also a constant buzzing from the insects. And every so often, as you're crawling along, going up and over the root system and under branches, there'll be a large splash. Now, this is usually just a branch falling in the water, or some of their seed pods, or maybe even one of the snails or large crabs falling off. But every so often, it might be something a little bit larger. It's also quite hot. When we're working up in the northern reaches, you're talking 45 degrees on a reef flat, and you've got the glare off the water. Well, you can't quite make it out, but this is our intrepid field leader, Clay Bryce, and he always wraps himself up in a in a tea towel, effectively, to keep his face out of the sun. Then we go back to the boat, or whatever we're calling a base for the trip, and then the sorting process, which takes a lot longer than the dive does. It's not as simple a process as it could be. For me, seeing as I'm pulling up bits of dead coral, seaweeds, rocks, I'm then hammering them open, trying to get what's inside. Dead coral's very porous. There's a lot of spaces inside that crustaceans like to hide in. Same with seaweeds, they tend to cling to it. I'll bring up sponges, and sponges have a lot of hollow space. And once again, crustaceans like to hide in it to keep away from such things as the potato cod. Once we go through the sorting, then we've got the preservation process, which can be a bit sad. We try to be as nice about it as possible. The smaller animals, it's fairly quick. Things like this pistol shrimp here, and larger animals we need to put into a bucket of ice. Pistol shrimp in particular tend to lose their pistol, which is this claw here, which hasn't quite turned out as much as I want. And basically this is, and in a large claw, they've got the smaller claw next to it, and basically they open it up and they form like a little trigger, which basically snaps when they shut. And underwater you can hear this. It's like a pistol shot, which is how they get their name. Pistol shrimp is just like underwater. And you put them in ethanol and they just go mad, and then it falls off, which is very sad, especially when you've got a lot of them all in the one jar and you think there's going to be about 6 or 7 species, and then all the claws fall off. It's a very important feature for using for the identification of that group. That ends the fun part of the trip. The data entry is necessary, but it's not why we became biologists. You spend untold hours in front of the computer trying to make sure that you put the data in correctly, put it in, check it again, and basically that then gets fed into our system, and we give them a label. Now, I've skipped the identification process. I didn't have any reasonable images that kind of illustrated it, so I'm going to talk about it with this slide instead. Basically, the identification process is the longest part of any survey, especially if it's a particularly successful survey. Basically, we've got to sit down, we get the specimens, and for me, coming from New Zealand, I'm not used to tropical zones. As Rachel said, my big biodiversity research was in the Southern Ocean, completely different, and deep water too, which doesn't help. So for me, I've got a lot to learn. It's going to take me quite a while to work through the specimens I've collected. And basically, as the taxonomic process goes through, groups get revised, the descriptions get tightened, you produce keys, guides to help other biologists to identify things. And basically, we've just you got your best guess what's going to be what it's likely to be. Go through one reference, you go, right, I think it might be that. Then you've got to find another reference to correlate, and then it can snowball, and you can spend hours trying to identify one specimen. But it's always handy when you go, that's it. Hey, I can move on to the next one and then spend another couple of hours. And people, well, even consultancies that ask us to do these identifications don't really understand the process involved. A lot of people do assume that everything's been found, or at least what hasn't been found is rather unimportant. But yeah, once that happens, once we get the identifications, if we can get the identifications, they go into the collection. Hopefully, with most of these trips, we are aiming to find new species, which is the basis of a lot of our research, is the description of new species, as well as then the revision of the groups that those species belong to, and it's a bit of a self-cleaning process as you go through. Well, that's the aim for it anyway. So then they get arranged into their families and species, and basically the collection then acts as a scientific library as well. Experts from around the world may disagree with your findings, want to see the specimens that you've said are something. They might get them, they might disagree with you again, say, no, you've got that wrong. It's one of the great pastimes of scientists is to prove each other wrong. They might come back as it being right. But yeah, first set of trips, I should say, Dampier Archipelago. Over 3 years, we surveyed it. It's a fairly large area that we went through, and we ended up with a lot of specimens. The Northwest Atolls, on the other hand, which I found here, that's Mermaid Reef there. That's part of the Rolly Shoals. And then there's Scott Reef, which is that dot there now. And just above that, which doesn't quite show up on the map, is Serangopatam Reef. These are in the Timor Sea. And this is what they look. They're nice coral reefs with a lagoon in the middle. As pristine as you can possibly get, I would have thought. Being so far offshore, they don't have a lot of terrestrial runoff like the Dampier Archipelago does. The inshore Kimberley is notoriously murky. But then, back to the Dampier. This is going to jump around a little bit. I'm hoping it's a bit clear, but it might not be. The Dampier results. For the collections, there were 2 dive expeditions and a dredging expedition. So the first 2, I think it was 2 dive expeditions, each lasting around 3 weeks. And then a dredging expedition in the deeper waters that we couldn't really reach with diving. So around the 50-meter mark. And then we had a workshop. Now this does require a little bit of explaining. Basically, seeing as space on the boat for the dive expeditions was limited, we couldn't take everybody that was interested in the area. The workshop, once again, I think would have been funded by Woodside. We actually invited people that worked on the groups that weren't represented during the actual field work to come in and actually do some work. We chartered smaller boats, take them out diving, shore collecting, and the like. And then with the express idea that they would then publish the results of what they found. And the identification process for all of this required 85 scientists from 25 countries. Now, one thing that people don't quite realize with this sort of system is that a lot of the identification process, like we get paid, or we would get funded, sorry, to do the expeditions. We might get some funding for consumables like glassware, ethanol, labels, label paper. Identifications are hardly ever paid for. A lot of those scientists did it off their own back. Sure, they were in positions where they had salaries, and the reason why they do take these sorts of jobs on for free is because it's actually in part of their research interest. A lot of scientists are actually in science because it's a hobby, and they like it so much that they'd probably do it even if they didn't get paid. And if you have a look at how many retired curators are still at the museum, you'd kind of get an understanding for that. I think Loazette Marsh, who was former curator of Echinoderms, after I think it's close to 20 years of retirement, still comes in twice a week and works on the Echinoderms. The dedication that is shown is amazing. You wouldn't get that in banking. They're out the door as soon as they can. But anyway, enough about them. So we found over 4,500 species identified. It's a large number, and it's hard to comprehend. I find it difficult to go, okay, so what does that actually mean? And I did search for some nice figures to compare with, like Ningaloo, but I couldn't find too many. We did find 268 species that had not been described. Records for Australia. Now this is the records is that it's never been published to have been found. It doesn't mean that it suddenly appeared. It means that it's either been missed in previous work, or just missed in previous work, or just misidentified, or just completely not even tried to be looked for. And so we learned a lot about the Dampier Archipelago because of this. I'll start with the corals because they're probably the easiest to explain. Prior to our work in the Dampier Archipelago, there were what are known as the crown of thorns surveys. The crown of thorns starfish, it's considered quite the pest for coral reefs. It's a very large, spiny starfish, many arms, and it creeps across coral reefs, eating the colonies. It was once considered to be the death of the Great Barrier Reef, largely due to fishing of its predators. There was a giant, what they call the Triton snail, which was the shell's about that big. It would eat the starfish, sea star. Sorry, I need to keep saying sea star. But since that's been such a prized item by shell collectors, the numbers have dropped. Starfish don't get eaten anymore, so there's more of them. And there was quite a lot of damage done to the Great Barrier Reef thanks to these. And that sparked the surveys over here as well, just to see what the extent might be. So 254 species of hard coral have been found in the Dampier Archipelago. We recorded 14 new species on top of what had been known through this crown of thorns. And compare that with North Atolls, roughly the same number. 20, obviously nobody, these are much more remote areas, harder to get to, so we had a few more records. No new species, but that doesn't mean they're not there. And one thing, we would have expected more species of hard coral at these atolls. One thing that got brought up when I did my practice run for this speech, for this talk, was that actually 2 years before we did this, Cyclone Faye ripped across the top of Scott Reef. And basically, the images have since been shown. There's a big satellite image of Cyclone Faye across the top of Scott Reef. And then the huge coral bommies, as they're known, they've been lifted up and put on top of the reef. And there's photos of various people just going, hey, and standing on top of the big boulder that's been lifted up on top of the reef. It's, we're talking several tons of rock lifted up by the cyclone and dumped high up on the shore, where it's still exposed at high tide. So it's not just that, you know, it's low tide, it's moved, it's rolled around a little bit. It's been picked up and dumped somewhere a significant distance. So that's had a serious impact on the diversity there. And you'll actually see that with some of these other numbers, is that you'll find that the coral's still quite high, but as I got told, is that they were really quite small colonies because of the regrowth. And as we go through, you'll see that some of these other species haven't quite bounced. So with the fish, there's always a lot of fish species. Fortunately, that blue spotted ray hasn't popped up quite so nicely. But now we've got a total of 650 species, fish species in Dampier. The Echinoderms, always a quite prominent member of communities. We found 286 species, but only 78 were previously recorded, despite the fact that they were doing a crown of thorns survey, which was looking at Echinoderms. They obviously were only looking for the crown of thorns. 4 new species and 6 Australian records, and 1 new record for the state. So now, in the crustaceans, once again, large numbers. It's hard to comprehend what these sorts of numbers can mean. But when you think about the total number of crustacean species in Australia, there's approximately 6,500, and that includes everything from your freshwater crayfish all the way down to the abyssal benthic copepods, which are minute. So having 10% of all crustacean species found in Dampier is still, it's quite it's an incredibly diverse area. I don't have a comparison with the Great Barrier Reef, unfortunately. And one thing I should have said with the corals is that it was 250 plus species on this side. Great Barrier Reef's got somewhere between 300 and 400. And considering the size of the Great Barrier Reef and comparing the size of that with Dampier, it kind of puts it in a perspective that, well, maybe the Great Barrier Reef isn't so good, it's just huge. But once again, new records, plenty to find. And you see the crustacean from the Northwest Atolls, what should have been a more pristine part of the country, actually turns out it's reasonably got a lower diversity. And one potential possibly is because the cyclone wiped out large amounts of coral reef, a lot of the crustacean species just didn't have the habitat that they needed anymore. Especially a lot of the there's a lot of little small shrimp that hide in the polyps of corals, the little feeding individuals inside the coral colonies. There's a lot of species that are only found on certain corals, certain anemones, things like that. So those sorts of things, once their habitat's gone, what are they going to do? Now, this is a quick diversion from those numbers before I get into something a little bit more serious. The Xanthidae, which is a family of crab, commonly called the black-fingered crabs, because the dactyls, well, the claws on the fingers of the claws are usually black, not always, unfortunately. But it's the most specious family of crabs in the world, and there's 168 species in Australia. In Dampier, we found 57, and in the Northwest Atolls trip, we found just over 40. Now, that's a considerable portion found once again at a very localized, reasonably localized spot. So there's no no surprises that there were a lot of them, but that there's so many of them is actually quite good. Another reason why I bring these guys up is that it's the only family with a poisonous crab. And they actually, well, this one here, which is the mosaic crab, actually contains tetrodotoxins. Now, this is very toxic. No known antidotes, 25 milligrams of which can kill an average-sized person. And if you've, well, I have to get reference to The Simpsons. If you've ever seen the episode where Homer Simpson goes to a Japanese restaurant and eats the Japanese fugu, which is a pufferfish, that's the toxin that has. And it's a Japanese delicacy, and if you don't cut it right and cook it properly, you kill the person who eats it. So it's for that just to be wandering around on the reef. If you're ever hungry, don't touch it. And that goes for the rest of the family. I wouldn't really risk it. I don't know about that species, but that one's the one on the other side, which is the shawl crab, because it's got a nice lacy pattern on the back the back of its carapace. Also poisonous. And there have been deaths recorded from people eating this species, not these species. I don't think it's these species that were eaten, so I don't know if they kept the carapace for identification. There have been deaths recorded, but yeah, just an interesting little story that I thought I should share. Now I'm getting into something that I'm going to start making some assessments on basically talking about the various groups. The mollusks. Mollusks are always a very diverse thing. You've got the nudibranchs, you've got the snails, you've got chitons, scallops, oysters, octopus, squid, cuttlefish. And despite the fact that mollusks are heavily collected for their shells, for their amateur collections, there's still new species all over the show. Now, using a nifty little Venn diagram showing that the species shared within the Northwest Atolls reefs. As you see, if the numbers add up to roughly 55% of species found between. You're always going to get species found at one site that you're not going to find at another, no matter how hard you search, there'll be something you miss. So 55 shared between these groups is quite a high number, especially considering if you look at the numbers shared between Scott Reef and Mermaid, which is, I think it's 450 kilometers apart, you've still got 200 species shared between the two of them. Now, keep that in mind about the 55%. Well, Tridacna, the giant clam, just before I launch into something else, I'll split off onto another tangent, was highly prized for as a collector's item. It's reasonably protected. I'm not too sure on its exact status, but one of the trips to the Northwest Atolls, it was actually looking at this population. The area is, I think it's considered native title for Indonesians to come down and fish there. They've been fishing them for generations. And the giant clams is an obvious source of meat, being so large. Their blue lips or green lips or brown lips sticking out of reefs are an incredibly beautiful sight to see when you're snorkeling or diving. Unfortunately, unfortunately the cyclone got in the way, and the population has dropped by a factor of 10 since the late 90s, which is very sad. Now, sponges. This is a group that's, for want of a better word, neglected. They've been always considered of having no economic value, and outside of the kitchen sponge, what uses are they other than decoration? Now, keeping that in mind, there are only 14 species that have ever been recorded at Dampier. We collected 275. 165, completely new. So, of course, 261 are new records for Western Australia. Now, comparing that with Northwest Atolls, it's the first records of sponges from the area ever. Nobody had ever collected them before. Not to say that they weren't there, just people were neglecting to actually collect them. Or if they did collect them, they just got put in a jar and nobody looked at them. So we found 32 species, and only 29 species could be fully identified. Now, I need to explain that point just a little bit. With the identification process, you are reliant on good literature or access to good specimens. With a lot of old literature, basically, when Linnaeus went about his Systema Naturae, he was the first one to set about or actually describe these things. So basically, he didn't know how many characters he needed to use to define particular groups. And he estimated that there was only about 15 to 20,000 species worldwide. We're now over 1 million. So as more specimens get pulled up, people look at them more. Obviously, the harder you look, the more you find, of course. So I can't say that the 103 species that couldn't be fully identified are all new. It's just that with the amount of effort that we could put into it, we just couldn't put a name to them. So the 29 species are named or previously named. So there's a potential of 103 new, but I can't give you that number. And a bit more about sponges. They're not just bland, encrusting animals. They're very surprisingly complex animals. They filter the water to feed off the plankton and bacteria and nutrients that are in the water column. And the statistic I've been given to say is that a tennis ball sized sponge can filter 5,000 litres of water in a day. And you can talk about, if you want to multiply that into Olympic sized swimming pools, it's going to be a lot. So they can be quite colourful. They also provide amazing habitats for every other animal that wants to hang around on them. And they're also really large. A lot of people don't think, kitchen sponge fits, but get one that size. It would have been really nice if she'd tried to fit into it, but I guess it might have split and that wouldn't have been as good a photo. She is a normal-sized person, before you ask. Now, with the sponges, as I said, being neglected, not a lot known. But one thing that was always common through is that one sponge, as soon as they hadn't been worked on particularly well, it was considered sponges. Well, they must be fairly widespread. But if you look at the numbers, those species were shared, well, 14 species were shared between Scott and Sarangapatan, as well as mermaids. So that's not very many were shared between those reefs in northwest Atolls, despite the fact that Sarangapatan and Scott are close to each other. It's what, about 25%? And then if I compare it with the Dampier Archipelago and another study that had been done earlier by a guy from John Hooper from Queensland Museum, you can actually see that 10% of species collected are actually shared at all. Is a remarkable uniqueness between these sites. So the Sahel Shelf Reefs is just a little bit closer to Indonesia and Timor, East Timor from Scott Reef and Sarangapatan, but very little is shared between these sponge communities. Now, that's remarkable, and if we're going to develop in these areas, we need to recognize that these areas are distinct from each other, rather than saying, well, we're going to just keep this one and we'll just bulldoze another one. And of course, the next question is that, well, with the summary is, basically, most sponge species are unique to a particular atoll. High rates of endemism. When we talk about endemism, we're talking about things not found anywhere else. And with, say, kangaroos, endemic, koala, endemic. Sponges, lots of endemics, not just one koala species. And that's a small bullet point. So it's important to protect each reef, or at least parts of each reef. With the Northwest Atolls, there was, I think they've given up on the idea, I'm not entirely sure exactly, but there was the idea of turning part of Scott Reef into a linking zone for some of the Timor Sea drilling sites. And basically the boats would come in and fill up. So they were going to put a big platform in the middle of one of the lagoons, which is one of the reasons why they sent us there, as well as another, well, I'm not too sure exactly how we chose the sites, but so protected areas, marine sanctuary zones, no drillings, no explosions, no dredging, things like that. Just need to be put in place. And with sponges, I chose the sponges because the numbers are huge in terms of what we don't know. With the mollusks, there's a lot of wide-ranging species, and same with the corals. There's a lot of wide-ranging species. Crustaceans are the same. Sponges, nobody knew that they could be that restricted. And of course, people are going to ask, well, what's the point of a sponge? I would argue that they're as important as the coral reef. As the corals kind of decline with depths, sponges take over. And you get these magnificent sponge gardens. And basically, they form the next step of habitat. And another reason is that the sponges are actually becoming quite important for the bioprospecting that I mentioned earlier. Effectively, because, well, whenever you see a sponge underwater, it's usually clean, clear by itself. You have any number of other organisms in the water, and they're covered in all sorts of crap. You get you put wharf jetties in, and within a month they're covered in barnacles, seaweeds, hydroids, all sorts of sessile animals. Sponges keep themselves very, very clean. The reason for that is that they've got an amazing chemical defense system. Much like we have symbiotic bacteria in our guts that help us digest, provide us with, I think it's vitamin K, and things like that we've got in our guts, and we need to keep them actually quite healthy, or else we get quite sick. And if you take antibiotics, you'll notice you get antibiotics and you start feeling terrible. Much the same with sponges. They've got little bacteria living inside them. A lot of them actually, surprisingly, have microalgae living inside, which photosynthesize and provide them with energy, much the same way as corals do, or some of the corals do. So when times are tough plankton-wise, they've got energy from the sun. Now, with the chemical defense systems and keeping their internal bacteria in line, this has developed a quite potent defense. Effectively, you don't get fouling organisms because they're producing all sorts of little toxins, and their immune system's going, oh, that's foreign, and I need to expel it. In fact, apart from things crawling on the outside like crabs or starfish, there's worms that will burrow, form tubes and burrow into sponges, and barnacles are about the only thing that foul them. And there's quite a lot of species of barnacles that foul them, but they're all one distinct group of barnacles. It's not just any old barnacle that you find attached to a wharf. There's a particular genus, effectively, of sponge barnacles that embed themselves into the sponge tissue where they gain protection from their own predators and somehow manage to subvert the chemical defenses of the sponge. And because of the chemical defense, or leading to this point, they've been targeted as potential anti-cancer agents. And with this growing economic interest on part of the pharmaceutical industry, the need to understand the species level diversity of sponges is incredibly important now, especially when it comes to ones that are now potentially limited to a very small atoll in the middle of the Indian Ocean. If somebody, I don't know if you've seen the movie Medicine Man with Sean Connery, where they're actually in the Amazon jungle and they've found something that's active against cancers and they couldn't figure out what it was. And it turns out to be the ants found on one tree, and then they knocked over the tree with the forestry guys knocked over the tree. Similar sort of thing. If you go, right, we need to build a wharf out onto this reef, knock out a sponge species that could potentially save people from cancer, it's going to look a bit bad. But with a general result for the Northwest Atolls, just completely getting away from the sponges now, Scott Reef we found was the most diverse of the Northwest Atolls. One thing I've learned in preparing this talk is that preparing summary statistics for something that you weren't a part of is quite tricky. It's all very dislocated in a lot of different areas, and you have to keep asking a lot of people over and over again, is this correct? But basically, Scott Reef is the largest of the reefs that we sampled, and it just had a larger range of habitat. Habitat complexity equals species diversity. If you've got a flat piece of rock, you'll get less than if you have a rock that's got some shape to it, some clefts, crevices, overhangs, caves, things like that. But now I'll give you a bit of a rundown on what we're currently doing with the Woodside Partnership. In October last year, we mounted the first part of an expedition to the Kimberleys, where we're looking at the differences with inshore and offshore areas of the Kimberleys. Basically, we went to Adele Island, which is a coral atoll with a spinifex grass, sandy island in the middle, huge bird population, lots of brown-footed boobies, frigate birds, and what was the other one? Some sooty terns or some of the birds, I'm not too sure. And then there was Montgomery Reef, which instead it's very different geologically. It's not a coral reef. It's effectively a part of the Kimberley that's sunk and now is exposed just off the coast, nestled in. I can't remember the area around it, unfortunately. In the middle of which there's a muddy mangrove swamp, which was very dense, and we didn't unfortunately get into it. It would have been really nice to have sampled that, but we had some very good reasons for not doing that. Now, in terms of scale, Montgomery Reef, obviously, basically that green part with the sandy bits around it is all that is exposed at high tide. With the king tides, you've got 10, 12 meter tidal difference, which is a staggering amount of water moving around. The rest of the reef gets exposed, and that's 300 square kilometers. That's bigger than Metro Perth. And of course, when we went there, it was like you get there, you couldn't see the mangroves from the edge of the reef. It was amazing. And the amount of water that has to come off during the tidal cycle meant that the top of the reef flat, so basically you got the mangrove swamp, and then it slowly slopes away, and then all of a sudden it just cuts off like that. It's not quite a cliff, but you've got all this, it's like an infinity pool. You look out across to the horizon, and it's just water, flat, calm, dead straight. And with the haze that we were having through some of the fires around at the time, you couldn't even see the horizon. It just went into a milkiness and then goes blue again with the sky. It's really, really quite nice. And then you get the edge of the reef, and it's always flowing with water. There's not enough time for the water on top of the reef to get off the reef before the tide comes back in. And you can see here there's a deep cut, and that's like a raging torrent at low tide. And it's really quite amazing. You get to the edge, and basically it slopes down and then kind of flattens out again and then goes straight down into these channels. And you get to a bit where there's a bit of a curve around and there's a bit of an eddy, and you see all the fish just sitting there. And basically they're just sitting there trying to stay out of the current. And you've got the turtles just cruising around in there. They know the fish are struggling to swim and stay upright. So they're just cruising around having a great time. As I said, we didn't go sampling at Montgomery Reef, thanks to one of our other former curators telling us that there was sightings of 3 6-meter long crocodiles in the area. So we didn't dive, but we still went trekking around on the reef, where it's probably just as dangerous. We didn't get into the mud or the mangroves because of that, but I never saw any of them. Apparently there were some sightings, but I didn't see any on the trip. I was very disappointed. And the health and safety, everybody knows it's going a little bit mad, but seeing the list of things that we had to worry about kind of made some of us go, well, maybe we shouldn't go. Crocodiles were first of all on the list. They were talking about the most aggressive during the breeding season, which is from May to October. Or it was like, okay, we're going in October. And then there were sharks to worry about, jellyfish, stonefish, blue-ringed octopus, strong currents, poor visibility, extreme heat, sunburn. It's a lot different from New Zealand, where there's nothing that will really do anything to you. Terrestrial or marine, you might get mauled by a seal, but you're not really worried about it. But anyway, preliminary results are indicating that we've got a new species of velvet fish. These guys are really cute. They basically mimic seaweed under the water. They just kind of, instead of fighting against currents, they just kind of roll with it. And you can see it's got a large dorsal fin down the side. Well, not the top, sorry. It's on its back, not the side. And basically that catches the current as the waves surge through, and it just rocks backwards and forwards, keeps pace, and it just looks like a drifting piece of seaweed. That one's a bit red, but I think it's starting to show some stress from us catching it and trying to take some photos of it. But underwater, it's quite a lot greener. And this guy's common name is going to be the bearded velvet fish, because it's got a strange little tuft, a little barbell thing underneath its chin. And you can kind of see it, but it's not quite in the photo. And a new fish is something is something to be quite, well, not surprised about, but it's quite something to get a new species of fish on a reasonably short trip. There are 120 mollusks, echinoderms, 190 corals. You'll notice that, I've gone too far again. You'll notice that the numbers are down. Basically, this is a 3-week trip. And basically the Kimberley being, the general trend for looking at what was in the water is that it was murky. The coral colonies were all stunted. There was a lot of encrusting corals, so just kind of going over the tops of rocks. Not a lot of branching corals where things can get in and hide. There was a fair amount of seaweed, but a lot of the sites were just a lot of coral rubble. So basically the habitat complexity was down compared to, say, the Northwest Atolls and Dampier. And because of the sediment in the water, basically you got all the silt coming down from the rivers, suspended in the water, blocks light. The corals that, basically, the coral reef obviously is based around corals. And with the low lights, quite a few of the species can't survive. So they're not there. And therefore, a lot of the crustacean species, a lot of the mollusks just aren't there. After the first couple of dives, everybody's going, oh, this is going to be a boring trip. There just wasn't that much for us to see. But in terms of Crustacea, I'm nowhere near coming up with an approximate total of how many species I've got, but there are 800 specimens. Well, that should be specimen lots. There's more than 800 specimens. And seeing as I'm new to the area, I've needed to rely on scientists outside of the museum. So I've sent them off to 13 scientists in 5 countries: the States, Germany, New Zealand, Poland. And unlike previous trips, museum trips where the focus has been on the decapod Crustacea, which are your more common groups, such as the crabs, shrimps, lobsters, I've also made the attempt at going for some of the microcrustaceans. The lesser known ones, they tend to be small, although some of them are actually really large. So trying to boost the output from the survey, seeing as there was a low diversity and a low density, I've gone for an extra method of collecting whatever's at the bottom of my sorting tray, sieving it, and then sorting it back under a microscope and picking out some of these guys. One of the reasons I wanted to do this is, well, strictly because in the museum I had a quick look before we went, and we've got maybe a couple of dozen records of any of these groups from that region. There's incredibly understudied in this part of the country, and as a result, I don't know anything about them, so they have to. Those are the main contributors to what's being sent overseas. And so in here we've got some groups you would recognise. We've got the amphipods, which are what you call beach fleas or sandhoppers, like this one here and that one. And basically, if you kick over some kelp in the high tide mark, you'll see things hopping around, and that's what these guys are. And then you've got the sea slaters or sea lice, depending on whether it's attached to a fish, such as this one. Much like the common slater in the back garden, but living in the sea. Basically, these are very diverse groups, and basically all of these ones are actually living in the sand, in amongst the sand grains. And much like topsoil erosion being a huge problem anywhere agriculture is being done, we can't create new topsoil. And in the marine environment, fortunately, there's wave action does its own ploughing action effectively. These guys help maintain sediment vitality effectively. They turn over the sediment themselves. Some of these other guys are burrowing, providing ways for oxygen to get into the sediment layers. So basically, if you've got a mill pond, sediment doesn't stir around, you end up getting a layer where all the microbes start to die off, and whatever else has been trapped in there dies off. And part of the decomposition process is that it uses up oxygen in the sediment, and so you get this anoxic layer. I don't know if you've got this problem in King George's Sound or not, but in New Zealand we had estuary areas where sea lettuce would wash ashore and just sit there and smother the top layer of sediment. And then as soon as the next storm came through, push it away, and all you're left with is this stinking, rotten what's underneath. Personally, I thought it served those people with the big houses on the hill right, but we ended up there. One of the places actually got guys with big tractors to go around and try and rake it up before it got to that point. But these guys are important to keep sediment health. Some of these groups aren't actually in the sediment. This guy here, this is commonly called a skeleton shrimp. You can see why it gets the term skeleton, but these guys tend to latch onto seaweeds and basically are ambush predators. And that one, well, you can't see it particularly well, but they usually have quite large forelimbs, which they just basically sit, wait for something to swim past, grab it. Whereas this one here, basically the ones with the big heads and the long tails, they sit in just in the lower top layer of sediment and turn over the little sand grains, cleaning off microalgae, feeding on whatever's deposited onto them. The one I particularly like is this little smiling fellow here. That's not actually its mouth. It's just the way the pigment patterns look. I'm eagerly awaiting what that's actually called. The common name is mussel shrimp or sea shrimp. Technically, it's known as the ostracods. They're an incredibly diverse group in their own, especially in the fossil record, and a lot of dating of sediments for prehistoric times is based around what species of ostracods are actually in it. And actually, there are more ostracods in more species of ostracods in the fossil record than there are living today, which kind of says that they once were much more abundant than they are now. But being so small, not normally bigger than your little fingernail, in fact, quite often much, much less, there could be still plenty to be found. Now, moving away from that, we've got the Northern Kimberley trip, which we're hoping to do this year in October. Same plans, hoping to get inshore and offshore. This time we're going to Cassini Island and Longreef. Cassini Island, I'm not too sure what sort of island that is, but it doesn't look like it's a coral atoll, whereas Longreef looks like it's a sandbar with a fringing coral reef. I've been promised the water is clearer and the animals more abundant, so I'm looking forward to that one greatly. And hopefully I'll at least made headway through the ones I've already collected. Then after that, well, we'll be producing a report, well, several reports hopefully, based on what we find, and these will be published through our museum records. Now, I don't like summary slides, so this one's not particularly good. I just want people to realize that there is a lot we need to learn. There's more species to discover, and we're just skimming the surface. We're in the top 20 meters of the water. Oceans cover what, 3/4 of the world's surface, and I wouldn't even want to talk about the volume of space that takes up in terms of when you start looking at the depth going down to 12,000 meters. The deep sea isn't the dead zone that it once was thought to be. And once we've got the species there, we need to learn why they're there. When it's not going on these surveys and saying we found so many species of something is well, it's quite good, but there needs to know more. Is that a unique factor? Is there something driving it? Is if we build a jetty somewhere to the north, is it going to all of a sudden disrupt currents and lift up a plume of sediments and then dump it on that area? There's a lot of extra thought that needs to go into the next step. We're just providing a baseline, a basis of information, and yeah, we're the first step. Then it's up to the rest of them to start working on it. And the next one is that there should be a role for industry. Industry doesn't need to be the enemy of the environment, like some people would like to make it out to be. Woodside has shown that active engagement with research can be fruitful. Yeah, we're not doing their work for them. We're not doing assessments of sites that they want to build an LNG plant on, and we're not making recommendations. And I personally would not ever want to make any sort of comment on what they're doing because of the money that's behind us, or not just because of the money behind us, but because that shouldn't necessarily be our role. If say with a topical issue being the LNG plant off James Price Point north of Broome, obviously you've got a whole suite of issues there, not just what's in the immediate vicinity. You've got what's known as the humpback superhighway, right? That goes straight past this LNG plant. There's things that need to be thought about. But yeah, industry does need to be involved at all stages of planning. Sometimes everybody knows that money talks, but unfortunately, sometimes you need to tell it to shut up. And that will bring me to the end of that. And I haven't named any names because there's just far too many to fit on this, other than Laura Roberts, who made sure that my slides were at least consistent. Thank you. Yes, sorry for going over time too.
Anybody like to ask a couple of questions? No. You weren't near within the area,
Mm-hmm.
extending out how far from the shore?
Not usually that far, because it depends on the depth, effectively. If we're diving, we need to stay around the 20-meter mark. We don't like going past 20 meters because you start running into issues with basically narcotizing the gas mix. Effectively, you need to start changing, and the level of qualifications for the divers need to increase and things like that. So if it's a large flat area, we go as far as we can. But normally, it also depends on the habitat that we're looking at. If it's a coral reef, once again, 20 meters is basically where things start to drop off again in terms of reef diversity. So usually that's, not usually that far from the coast. Sometimes half a K, sometimes maybe 2 or 3, 4 or 5, depends on the reef. Ningaloo Reef comes within 500 meters of the shore at Coral Bay, but I think it extends out to like 10 kilometers off the shore at some point, but I'm not entirely sure.
Now, the other thing was,
Mm.
what are there other factors out in the larger ocean areas that affect your work and what you do? You mentioned that there was the cyclones, but
Mm-hmm.
is there any problem with other things like shipping that's carrying load of stuff? I didn't mean just oil spills, I meant
No,
other things.
definitely. There's quite a bit of money spent. Basically, this comes under the biosecurity and introduced pests. So there's quite a lot of money spent keeping an eye on what's coming in on the bottoms of boats. And I've actually been quite surprised with how proactive Australia's been when it comes to the hull fouling. Basically, ships can be just floating reefs, very diverse. You get all sorts of things, crabs, snails hanging off the bottom of boats. And when they've got little nooks and crannies where the current, when they're out of the current from the movement of the boat, then you get fully adult breeding populations of things. And basically, since working here, I've had quite a lot of specimens come through from Sinclair Nightmares, URS Corp, some environmental consultants, bringing in barnacles for me to identify. And basically, there's a few species that are on what's called the trigger list. And basically, if something gets surveyed here by them that's got, say, one of the trigger species is the Asian green mussel. Not to be confused with the New Zealand green lip mussel, but this Asian green mussel, which they are very worried about that coming down and smothering all other mussel species. The boat goes back to wherever it came from. And that's a huge amount of loss for the company that wants the boat. Even just sitting in port, that's costing them tens of thousands of dollars a day if they're not doing something. And for them to send them back to Singapore to get cleaned, that's huge loss. And that's actually surprised me that that's actually made it into regulations. In New Zealand, when I was working on a similar project, we had an oil rig that was working off the west coast of the North Island, and basically it was going back to Port Phillip Bay to get refurbished. And AQUIS, the Australian Quarantine something service, I forget what the I stands for, inspection, that must be it. They said, right, we want you to check the pylons. And they came back saying, oh yeah, there's a green mussel on it. And then AQUIS talked to our version, the New Zealand equivalent, Biosecurity New Zealand, and they went, ooh, I wonder if it's ours or whether it's this Asian green mussel, which it's been in the country for 5 years already. I was thinking, well, quite clearly it's going to be our green mussel if it's going to be any of them. But they collected some samples and brought them in, along with some associated animals. There's some sponges, some barnacles came in for me, and there was a couple of crabs. The crabs turned out to be some wide-ranging species that's found in Australia, New Zealand, and some Polynesian islands and things like that. For it to be found that far south was a bit surprising, but not so much. But basically, we didn't get told the full story of where this oil rig had been. And basically, we got the identifications back. The barnacles I got, there were South African and Australian barnacles that they collected, as well as New Zealand barnacles. And that was like, ooh, good for me. And we told them about this, and then we got some information back from about some of the sponges that they collected as well. And basically, we couldn't get a name on them because sponges aren't particularly well understood. But our sponge experts said they're most comparable to a South African species. And we're going, ooh, this is going to be actually quite an interesting problem. Because what do? They can't go back to Australia until they've cleaned it. We don't want them to clean it in our waters. And then the mussels came back. And sure enough, there was the standard blue mussel, and there was also our green lip mussel. But then there was also what's the South African green mussel, and the South African ribbed mussel. And we went, oh. It was like, oh, they've actually found something that's actually. So like, with the biosecurity thing, a lot of the introduced species that we'd come across, it was all quite small critters or things that you wouldn't immediately associate as being potentially damaging. And this was the first kind of like, okay, our mussel industry is incredibly important. And if we're hocking off what's considered, in terms of mussel industry, an inferior quality product under our name, it's quite a bit of egg on our face, on the well, at least the mussel industry's face. So New Zealand tried to say, right, you can't clean in shallow water. You have to go out to 200 meters, clean there, and hopefully everything drops to 200 meters and doesn't do anything, and it doesn't survive. ignoring the fact that the oil rig had actually been in the country for 5 years already. And at this point, we found out, well, your oil rig's been in South Africa. Why didn't you tell us this to begin with? I was like, oh, yeah, we probably should have told you that. But basically, when they were going to clean it, they started cleaning it. Then bad weather moved in. And basically, an oil rig is the same as a ship. The captain's word is the final say. That he's got the safety of his crew as well as the rig to think of. They moved into Tasman Bay, which is near where I grew up. Well, it's the bay where I did grow up, actually. Moved in closer inshore. They still stayed 12 nautical miles off the shore, but the bay is so large and flat that it was actually in 40 meters of water still, which is an annoying depth for anything. It's too deep for standard diving, and it's just a bit, the amount of space that this thing takes up. They cleaned it all in 40 meters of water, and it's like, how are you going to clean it up? Because you don't have unknown numbers of the South African mussel, which 40 meters is not too deep for a mussel. And if that spawns, just around the corner of one of the other bays is one of the major spat collecting areas for the New Zealand green lip mussel. And all of a sudden, industry was like, well, we're going to have to call off our spat collecting this year, and the next year, and the year after that. And it was like, and all of a sudden, the industry was panicking. And basically, for Australia to be able to say, nope, you go somewhere else, and for it to actually happen, surprised the hell out of me. And very good, and very proactive of them. And I hope that wasn't too long-winded. Thanks, Ray.
Can I just say thank you, Andrew, for that? I sometimes am so busy doing other museum things that I actually get to miss out on catching up with the work that a lot of our curators are doing, and I love having them down so that they can fill me in as well. I'm sure everybody got a lot of information out of that they're going to go away with in terms of, you know, the fact that you're calling the baseline for speciation, and also probably fixing some of the problems with maybe the overzealous splitting of species in the past.
Hopefully.
Rectifying all that
yes,
stuff.
rectifying. And
yeah, look forward to hearing what happens from your
yes.
trip in October of this year. If everyone would like to just put their heads together.
Thank you. That's right.